So we are now studying DNA replication, what machinery and what enzymes we have to use. So first of all, main line come here. The enzyme which is required for DNA replication is DNA polymerase. And main thing is this, ki replication will not start randomly from any point. So replication does not initiate randomly at any place. It will not initiate randomly at any place. There is definite region from where the replication will start. And that is known as origin of replication. Origin of replication is actually, it is a point from where the replication will start. It is because of the requirement of origin of replication, we are using the plasmid in the genetic engineering. So see here, come in this diagram. If you check in this diagram, you will find E. coli, this is bacteria, and bacteria has only one oocyte. And this is eukaryotic chromosome, which contain 1, 2, and 3 oocytes. So it means, first basic difference you should know, replication will start from the oocyte, and prokaryote have only one oocyte, and eukaryote have many oocytes. Now, oocyte is a site from where the replication will start. It means DNA will go for unwinding from here. And when it goes for unwinding, check, it is making like a bubble-like structure. This is known as replication bubble. This is known as replication bubble. And this point is known as replication fork. So if suppose I assume only this part, in my diagram, if I assume only this part, means I am drawing it only this part. I'm assuming only this part because this was the oocyte. So I'm assuming only this part. Just see, once again, I'm rubbing it. Please focus the diagram carefully. I'm using different color. Just see, I'm taking only this part, half part. And this is replication fork, means this point is replication fork. See in our NCERT, this is the diagram, replication fork. Clear? So we have to first understand that replication will not start randomly. It will start from the oocyte. This is the site from where replication will start. Unwinding of the DNA will be done and replication bubble is made. And this point is known as replication fork. I hope you are getting it. Fork-like diagram, so we say replication fork. Now, during which phase of cell cycle the DNA replicate? You know very well, replication occurs during S phase of cell cycle. It occurs during S phase of cell cycle. Now, the main enzyme, now come here. The way I'm moving, please learn this way only. Though you require a set of an enzyme, it is not, it is only with one enzyme. E. coli for replication require a set of an enzyme. But which is the main enzyme? Main enzyme is DNA-dependent DNA polymerase. Because it uses DNA as a template, it will use DNA as a template. That's why we say DNA-dependent. And it will add more and more nucleotides. So it uses DNA as a template to catalyze polymerization of deoxyribonucleotide. So which is the main enzyme? Main enzyme is DNA-dependent DNA polymerase. And main thing is this enzyme is very efficient because if you see, it can do the catalyzation of large number of nucleotides in a short time. For example, E. coli, which has 4.6 into 10 power 6 base pair, can complete the replication within 18 minutes only. It means speed is 2000 base pair per second. The first thing is this enzyme will do the catalyzation at very fast speed. So it will do the catalyzation for the polymerization in a very short time. The time shortage is there. Second is it will do the polymerization with high degree of accuracy. So not only it is fast, it is accurate also. If any mistake is done during replication, we know it can lead to mutation. So it has to be very accurate and very fast also. Now, replication is energy expensive process. Replication is energy expensive process. So we want ATP. So what we are doing it, You know very well, we have studied sugar with one phosphate and one nitrogenous base, let us assume adenine. We say this is nucleotide. Nucleotide. And sugar with nitrogenous base is known as nucleoside. You know this. We have studied already in the starting कि nucleoside means sugar and nitrogenous base, nucleotide means all three. But if suppose I add 3 phosphate, 1, 2, and 3, so what will I say? I will say deoxyribonucleoside triphosphate. Deoxyribonucleoside triphosphate because I'm using 3 phosphate. Triphosphate. So this is the one which is actually very, 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 very important. Understand carefully. It is doing two function. What two function? It act as a substrate. It act as a substrate because it will be only added and it provide energy for polymerization. Now let me show you from another book how. See here. This is DNA nucleotide triphosphate. Clear? This is sugar, this is nitrogenous base and triphosphate because two terminal phosphate will be released and it is helping in making diester bond as well as hydrogen bond. So DNA, main enzyme which is DNA dependent DNA polymerase will do polymerization and it use deoxyribonucleoside triphosphate as a substrate. Now main thing is this, again come down, please come down and see. The enzyme cannot initiate the process. Enzyme cannot initiate the process on its own. That is why if you see in this diagram, we were having a short primer. This is a short primer because DNA polymerase can only add to the third OH group. So DNA polymerase cannot initiate on its own. It can only add on the existing strand. And existing strand is made by RNA. This is short RNA primer and third OH. So they are able to add. Clear? Now I'll just show you. Come here. See, this is the primase enzyme. Please focus. This is the primase enzyme, which is making the primer. And then DNA polymerase is able to do the polymerization. So let me show you once again this diagram. See here. So main enzymes, what we have used, main is DNA dependent DNA polymerase, but other enzyme first is helicase, which will unwind and separate the parent DNA. Then single strand binding protein to make it single strand, to stabilize it. Then we are using topoisomerase to break and again rejoin the parental DNA ahead of replication fork. This is replication fork, you know very well already this diagram. And primase to make RNA by using parental DNA as a template so that the DNA polymerase can add nucleotide. Because in our NCERT it is there, DNA polymerase on their own cannot initiate the process. So not only you require DNA dependent, so in addition to the DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication. So what are the additional enzymes? We studied helicase. Okay, let me show you helicase. We studied single strand binding protein. We studied topoisomerase, primase. These are the few enzymes. Now, when they are opening the DNA, main thing is this key, the two strands, because they are anti-parallel and DNA polymerase can do polymerization only from 5' to 3'. Again, complexity come. Let me show you the line. See this line carefully. DNA dependent DNA polymerase can do polymerization only in one direction, that is 5' to 3' only. It means again some problem came. So now let me explain you on this board. Check. Come here and understand this diagram. See, this is your DNA. We have opened this DNA. This is 3 dash, 5 dash. It means new 5 dash will come here and new 3 dash will come here. So I have to add short RNA at the new 5 dash. Now here I have added short RNA and here I have to add short RNA this side. This is the short RNA which is added, and now DNA polymerase will add nucleotide. The nucleotide is added. Clear? This much work is done. Now I'll open more because I cannot open it long because it's an energy expensive process. So this is primer. First time work is done this much. This is able to see single strand, it can move. Problem again come here, again I have to add primer. Let me explain you one more. Open more. First time primer, this is first time, second time. First time work done, second time work done. First time work done, second time work done. This can see, it can move forward, third time. It cannot see, again it will come back and it will add. So now black color is parent. Parent means template. So can I say template which is running from 3 dash to 5 dash is doing continuously replication? It will do continuous replication and it will produce leading strand. Am I right? Is it comfortable up till here?
Yes, ma'am.
And template, which is doing from 5 dash to 3 dash, will do discontinuous replication. Will do discontinuous replication. Not continuous, it's a discontinuous replication. And because of which they produce lagging strand and produce lagging strand. And the pieces which they are making, this is one piece, they made this one piece. What is this piece known as? This piece is known as Okazaki fragment. This piece is known as Okazaki fragment. Later on, what will happen? Another enzyme will come to remove the primase. It will remove. It will remove. It will remove the primase so that DNA polymerase will move further. It will move further and gap which is left out, that will be sealed with the help of DNA ligase. DNA ligase will seal the gap. DNA ligase will seal the gap. I have to remove the from here also. And this is the way how the working is done. See now next. Template with polarity 3 dash to 5 dash will do replication continuous. I think so I wrote this only. Template with polarity 3 dash to 5 dash will do continuous replication. Check once again. And template with 5 dash to 3 dash will do discontinuous replication. And in discontinuous, later on the fragments are joined by enzyme DNA ligase. So 5 dash to 3 dash discontinuous replication and they are joined by the enzyme DNA ligase. So already I told you cannot open at a stretch. No, it's very difficult. Why? Because it is highly energy process. So it will work only in a small segment that is known as replication fork. Now for your exam, how to learn? Please from here 3 dash. So 5 dash if you see continuously working. But if 5 dash, then it is coming out. Check it is coming out. I think so you match with my diagram, you will find this one right. In 5 dash, it is continuously coming out. Arrow is continuously coming out. So in this video today, I have completed the replication also. So just read the line carefully. Still, if any doubt, then please ask. Okay.